Assalamualaikum and good morning everyone. So today we are going to discuss about protein metabolism, alright, as well as the structure of kidney. Okay, so for STPM level, alright, the protein metabolism that we are going to discuss is about the transamination and deamination process that occurs when there is an excess of protein in the diet of an individual. Okay, so transamination, alright. So what happens in transamination, alright? First things first, the excess protein in the diet must be digested or broken down into amino acids, right? Because you know amino acids is the monomer for protein. So the protein, excess protein must be broken down first into amino acids, right? So these excess amino acids will undergo transamination process, all right? Now, transamination is a process where the amino group, right? The amino group of an amino acid is transferred, right, to the keto acid to produce new non-essential amino acids that is needed by the body, right? Can you get that? Right? Transamination is a process where this amino group from one amino acid, right, is transferred to the keto acids to form or to produce non-essential amino acids, okay, needed by the body. So apparently this amino group will be transferred here, right? The amino group from this amino acid will be transferred to the keto acid here, and this oxygen in the keto acid will be shifted here. Right, now to balance up the charges, the transfer of this amino acid to this keto acid comes together with this hydrogen ion. Okay, so this is the non-essential amino acids, right? So the amino acid A, all right, okay, that consists of the COOH and the R, okay, the R1 become a keto acid A. Okay, so this transamination occurs in the liver, right, and catalyzes by transaminase. Okay, example of transamination is the transamination of alanine, right? This is alanine. Transamination of alanine by transaminase, right, will produce pyruvate or pyruvic acid and glutamate or glutamic acid. Can you see how this amino group from alanine transfer to the alpha ketoglutarate, which a keto acid, right, NH2 will be transferred here, and this is what you get, glutamate, right? And then the oxygen from the alpha ketoglutarate will shifted here in the alanine, so you get this is what you get, pyruvate. Right. Okay, now deamination. So first things first, when you explain about transamination or deamination, you must explain that the presence of the excess amino acid, right? So the excess protein in the diet will be digested or broken down into amino acids. Okay. Okay, so the excess protein in the diet, first things first, will be digested or broken down into amino acids, right? And these amino acids will be channeled into the liver mitochondria. Okay, so the excess amino acids undergoes deamination process. Now, what is deamination? Deamination is a process where the amino group, this is one example of an amino acid, right? So in deamination, this amino group, right, will be removed and keto acid will be produced as byproduct. Okay, so apparently this will become the keto acid. Okay, now what happens to the amino group that was removed from the amino acid? So the amino group, okay, will combine with hydrogen atom to form ammonia. So we have ammonia here, all right, we have ammonia here. Initially this was the amino group which combine with the hydrogen atom to form an ammonia. Okay, and this ammonia, right, as we know, is very toxic, right, inside the human body, so it needs to be removed. All right, and this ammonia will be removed, all right, by converted first into urea, right, via the urea cycle or ornithine cycle, which occurs in the liver cytoplasm. So the urea will be excreted in the urine. So from this diagram here, you can see how this ammonia, all right, enters the ornithine cycle. Okay, ammonia combines with carbon dioxide and water, right, and then it forms the carbonyl phosphate, enters the ornithine cycle, right, and from the ornithine cycle, fumarate is being produced as a byproduct, okay, of the ornithine cycle, and this fumarate will enter the Krebs cycle for cellular respiration. You know, once this fumarate enters the Krebs cycle, the outcome will be pyruvate, right, and from pyruvate it can undergo glycogenolysis, right, glucogenesis, right. Okay, and then towards the end of the ornithine cycle, urea will be produced or synthesized and excreted from the body through the kidney, right, as urine. Now, what happened to this keto acid? Okay, this keto acid will be converted into acetyl coenzyme A, and then that acetyl coenzyme A will enter the Krebs cycle for cellular respiration. Okay, the organic acid here refers to the keto acids. Okay, and then this keto acid, right, apart from converted into acetyl coenzyme A, right, this keto acid also can be converted into glucose or glycogen, right, via gluconeogenesis. Okay, now the keto acid also can be converted into triacylglycerol as storage in fat cells. So these are the outcome of excess amino acid through deamination process. Okay, so now that's about it on protein metabolism. Okay, now we continue with Osmo Regulation. Okay, now Osmo Regulation is the regulation of the concentration of salt and water in the body, and it occurs in the kidney, right. So the principal function of the kidneys, right, is to help maintain homeostasis by regulating fluid balance and excreting metabolic waste. And the kidney also produce enzyme, right, which is renin, that helps to regulate the fluid balance and blood pressure inside the body. Now, let's take a closer look on the kidney, right. Kidney, right, has nephrons, right, many nephrons. This is one nephron. Okay, so a nephron is the physiological unit of the kidney that is used for filtration of blood and reabsorption and secretion of substances, right. What type of substances are being reabsorbed and secreted here, we will further discuss in the next discussion, right. Okay, now a nephron, right, this nephron here, as you can see, consists of glomerulus. This is the glomerulus, right, which is a cluster of blood capillaries that's surrounded by the Bowman's capsule. So this is the Bowman's capsule, and inside the Bowman's capsule is the glomerulus, right. Now the Bowman's capsule, or also known as glomerular capsule, right, is a structure that opens into long coil renal tubule. Okay, so Bowman's capsule is a structure that opens into a long coil renal tubule. Now do bear in mind that a renal corpuscle consists of this glomerulus and this Bowman's capsule. So these two structures will make up one renal corpuscle. Okay. Now this renal tubule, right, that I mentioned earlier. Okay, it's a structure that consists of proximal convoluted tubule and then loop of Henle and the distal convoluted tubule. Okay, how do you differentiate between a distal convoluted tubule with a proximal convoluted tubule? Very easy. Proximal means dekat, near. Distal means distant, jauh. Okay, so this proximal convoluted tubule is the tubule or renal tubule that is near to the renal corpuscle, while the distal convoluted tubule, right, is the tubule or renal tubule that is far from the renal corpuscle. Okay, you can see here in the loop of Henle we have the descending limb and we have the ascending limb, right? Descending limb and ascending limb. A closer look on the descending limb, we have the thick descending limb and we have the thin descending limb. Okay, and then on the ascending limb we have the thin ascending limb and the thick ascending limb. Okay, and you can see that these proximal convoluted tubules are surrounded by these peritubular capillaries. The same goes for the distal convoluted tubule, right? They are surrounded by peritubular capillaries, while this loop of Henle are surrounded by this vasa recta. Okay, there are two types of nephron, right? Cortical nephron and juxtamedullary nephron. Okay, this is how they look like. Okay, this is the cortical nephron and this is the juxtamedullary. The difference is for the cortical nephron, alright, this nephron located almost entirely within the cortex. You can see the renal, this is the renal cortex. This is where mostly the cortical nephron are located, right? And then this cortical nephron has shorter loop of Henle, and smaller glomerulus, right? Now a closer look on the juxtamedullary nephron. All right, this nephron located within the cortex, right, with longer loop of Henle that extend deep into the medulla. You can see the renal medulla here, right, with the long extension of the loop of Henle from the juxtamedullary nephron. Now this is important in concentrating the urine. Okay, later you will learn that this loop of Henle involves in the reabsorption of water, okay, into the renal medulla. The water is needed by our body, right? And then this juxtamedullary nephron also has bigger glomeruli or glomerulus. So blood flows, right, from the renal artery, you can see from here, renal artery, okay, to the afferent arteriole, then to the glomerular capillaries, okay, and then the blood will flows into the efferent arteriole that delivers blood into the peritubular capillaries and vasa recta. Blood leaves the kidney through the renal vein. You can see the renal vein here. So that's it for today. Okay, we continue in the next lesson.